Okay, good afternoon, everyone. Thank you. Welcome, everyone. It's wonderful to see friends and colleagues from around the world. My name is Chris Booth. I'm an oncologist in Kingston. And I just want to extend a very warm welcome to the CSO leadership group that's gathering here in person in Montreal. And we've arranged for a very stereotypical snowstorm in the middle of April in Canada, and to recognize the extensive CSO community that's joining online to listen to the lecture today. We'll hear a little bit more later today about the workshop that we just had with thought leaders, editors-in-chief of the major journals in our field, leading trialists from around the world, regulators, as we talked about some of the really important pressing issues in our field. I just want to acknowledge the Ontario Institute for Cancer Research has been instrumental in supporting the activities of today and this meeting. So I'm going to give just a very brief background, the origins of Common Sense Oncology. I think most people here in the room were also at the Kingston planning meeting 2 years ago. But essentially, this initiative was launched with two fundamental concerns, which is that many or most people in some health systems do not have access to those cancer treatments which make a real difference in their lives. And also, we had some concerns that in some contexts, the bar for approval and adopting new therapies may be dropping low, and we should aim higher and try to achieve better outcomes. And so why did we launch this grassroots initiative? I think it came from a place of caring for patients and families over many years, a recognition that we can do better. We also want to advocate for the things that we do very well and ensure that patients, regardless of where they live, have access to those therapies. But we were unwilling to remain silent on some of the problems in our field. But we didn't just want to be critics. It's somewhat easy to criticize. It's much more difficult to create solutions. And that was part of the ethos of this group from day one, is that we'll be willing to have uncomfortable conversations and identify things that are problematic, but we will work collaboratively and with an open mind to try to achieve solutions. We want to set an example for the next generation in our field, and really to create a place where we can have these conversations. I think all of us have recognized some of these challenges for many years, but we want to bring these out into a place where we can have honest discussion, show humility, self-reflection, and ultimately drive things forward to improve things for patients and their families. So going through the history of CSO, the initial leadership meeting was held now 2 years ago in Kingston. And the key questions that we addressed then were, what are some of the biggest challenges in our field? What are those challenges that we have some agency over and that we want to tackle? What are the guiding principles of this group? And how will we organize this work? And so this is a cast of characters that gathered in a small university town in Eastern Ontario a couple of years ago. And we had vigorous debate for a few days and mapped out an ambitious plan and strategy for the foreseeable future. And this culminated with the Common Sense Oncology Manifesto published in Lancet Oncology now about 2 years ago. And the mission of this initiative is to ensure that cancer care focuses on the outcomes that matter to our patients. The vision is that regardless of where someone lives, they have access to those treatments that make a real difference. We have a series of guiding principles that really determine and outline all the steps forward. We believe that cancer care is a fundamental human right, that patient and societal needs should be driving our research ecosystem and policy decision, that all of these issues require patient involvement and public involvement. We think that we need to aim high, and patients should be able to expect that the treatments we offer will help them live longer and better lives. Obviously, all of these discussions are grounded in the principles of shared decision-making, empowering patients to make the decisions that are best for them. Equity and access to care is just as important as accessing new innovation. So ensuring that patients have access to the treatment we already know works requires investment and health systems to mobilize. And finally, recognizing that comprehensive cancer care involves more than just medicines, and we need to advocate for all the other core services within our field. We've organized ourselves around 3 core pillars of work. We seek to improve how we generate evidence, how we interpret evidence, and how we communicate evidence. The generation of evidence is largely focusing on how we can improve and work with our partners in clinical trials to ensure that we identify the best treatments for patients in the future. Evidence interpretation is an educational bucket to improve critical thinking and skills and critical appraisal amongst oncologists. And evidence communication spans the continuum from the bedside, doctor-patient communication, how we can talk about these difficult issues and inform patients in a clear and compassionate way, to make the decisions that are best for them, but how we talk about things at a macro level, both within our own community and more broadly with public and policy makers. We've organized ourselves into a group of projects teams that have a co-leadership structure, and from day one, patients, patient advocates, and people with lived experience with cancer were embedded in all of these project teams, and in fact hold leadership roles as well. And we're going to hear more over the next couple of days about where these project groups have come over the last 2 years. We also have a lot of excitement around the Common Sense Oncology Fellowship, which has grown now to include a very strong and rich partnership with EORTC, who shares a lot of the vision with CSO. We've been very privileged to work carefully with that group to design a number of projects that we'll be discussing over the next couple of days. There's been a lot of discussion about, is ice fishing really important in Common Sense Oncology? And in fact, sometimes people don't understand my sense of humor and think I'm talking about a real entity. And I can tell you that there is at least 3 people in this room, including someone who will remain unnamed, but an incoming CSO fellow from a major academic center in Australia, who asked me last night, Chris, are we really going ice fishing? There was no ice in the St. Lawrence River. And so, in fact, we're not going ice fishing, but we did arrange for a blizzard to come in to welcome you to the great white north of Canada. The leadership meeting last year in Montreal, we tackled a number of other issues. How to accommodate the explosive interest we experienced with CSO in year 1, how to build capacity, what are our strategic priorities, how do we align our work plan with these priorities, and how can we develop a communication strategy to advance this work? And so this is the cast of characters last year from Montreal. And this led to a lot of work over the last year to design our strategic plan. And very briefly, we have 2 strategic foci. We want to focus on ensuring equitable and accessible cancer care, and we also want to ensure that treatments in the research ecosystem are oriented around outcomes that matter. We had to have more focus. That always became clear after year 1. And so our strategic goals are the design, analysis, and reporting of cancer clinical trials are focused on outcomes that matter. Number 2, clinical education incorporates common sense oncology, principles, and competencies. Number 3, doctor-patient communication informs and empowers patients to make treatment decisions. And number 4, cancer medicine policy aligns with and advances outcomes that matter. Ultimately, what we're hoping to do is lead to cultural, institutional, and individual adoption of CSO principles so that we improve equity, access, and patient outcomes. And here we find ourselves at our 3rd annual leadership meeting. And we're going to talk over the next few days about these questions. How do we leverage and implement the early outputs from our project teams? How do we expand our reach? How do we better engage our members and partners? How do we refine our communication strategy? And how to harness the fact that clearly in the last number of years, some of these ideas are becoming more mainstream and becoming more discussed at meetings and in journals and in other outlets around the world. So just briefly to walk through some of the work that we've done over the last year. The RCT project team, co-led by Ian Tannick and Bishal Gyawali, have published the Common Sense Oncology RCT checklist. There is another major project almost completed, a joint venture between leadership at EORTC and Common Sense Oncology, principles for design and incorporation and reporting of quality of life in clinical trials. We recently have a paper led by Dr. Eisenhauer, which is a guidance for discussants at conferences. And there's work underway to come up with Common Sense Oncology principles for single-arm trials and how to approach designing new treatments for rare cancers. The Patient Priorities Project team, co-led by Michelle Tregear and Chris Jackson, have been very busy bringing to fruition and completion a patient framework for clinical trials. And this has involved extensive engagement with a speaking and listening tour with patient advocacy groups around the world that has deeply informed our work in this space. There's another project well underway, about to be launched, coming up working together with patients and their families around the world to understand which questions do they think are the most important that need to be addressed in a consultation. And we have evolving projects looking at how we can improve bedside communications and ultimately work with the educational team to improve oncologists' understanding of these nuanced issues. The education project team, co-led by Scott Barry and Heide Verduzco Aguirre, have done an extensive review of medical oncology educational curricula around the world to identify competencies and principles of common sense oncology that could be expanded and celebrated in other health systems. And now they're undertaking a Delphi process to develop new curricular modules to foster these skills in critical appraisal, epidemiology, communication skills, and health policy. There's been a number of common sense oncology journal clubs and webinars, outreach events held at our major meetings around the world, and the development underway of a common sense oncology resource library for the broad community. And the Health Policy Project team, co-led by Manju Sengar and Brooke Wilson, have done a global engagement exercise, again, a speaking and listening tour with oncologists around the world. They're doing work now to understand the barriers and access on the ground of essential cancer medicine in low-resource health systems. There's work right now understanding regulatory approvals and decision-making when benefit is uncertain, and physician insights about some of the complex issues about endpoints and effect size. And there's a study about to be launched to see if we can improve and better support patients and families when they are making complex treatment decisions in the last year of life. This is the recent publication led by Bishal and Ian and the rest of the RCT project team that has done really a very good job of outlining a series of concepts, both in the design and the reporting of clinical trials, that we think will be hopefully a useful starting point to raise the bar on quality and outcomes, asking important questions about trial representation of clinical practice, the design elements regarding endpoints, effect sizes, analytic considerations, including informative censoring, follow-up for survival, clear reporting, and ultimately ensuring that the manuscript reflects what a patient can expect if they were to accept this treatment. The checklist also seeks to empower information for both clinicians and patients and families. Part of this manuscript talks about an abstract having a clear definition of the primary endpoint, reporting both the hazard ratio as well as the absolute magnitude of benefit, an objective summary of toxicity and quality of life, and also within the primary publication, a short summary for patients and families underneath the abstract so they can have an understanding of what this would really mean for them. We've had remarkable engagement with really institutions and organizations around the world. We were very fortunate to have a dedicated educational session last year at the annual meeting at ASCO, where we focused on common sense oncology and had broad discussion with stakeholders. And as mentioned, Elizabeth has led, together with the rest of the RCT team, guidance for discussants of RCTs at major cancer meetings. And as part of this engagement, speaking, and listening exercise, we've really been privileged to be invited to give invited talks, seminars, have meetings to talk to the major decision makers, bodies, and agencies within our field to learn how we can move things forward. At this point, I'm going to make an announcement about CSO that not many people know about, but our story for the last 2 years has been so interesting that we've been approached to create a documentary about common sense oncology. This may or may not be true. What I can tell you is that everyone in this room will be played by a famous movie star from Hollywood. But the Netflix producer was very clear. There is one person who will play themselves. And that's because there is no one more handsome or more smart in Hollywood than Dr. Bishal Gyawali. So please stay tuned for this important development in the history of common sense oncology. Where do we look forward over the next year? Some of the key priorities that we'll be talking about over the next couple of days. A communication strategy and implementation, doing a better job with member resources, a website redesign, communications both internally and externally, working with media to tell the story in the public space, and the thought leadership in each of those domains. We want to better engage and grow our membership, more stakeholder and partnerships, discussions around the world, and there's discussions, and probably stay tuned on this for announcements regarding the national and regional hub model that we hope to roll out very soon for common sense oncology. So in closing, I just wanted to offer a few thoughts about what I think makes this initiative special, and I think what brings all of us back to these meetings, and has created a huge amount of interest and energy and collaboration in the space, which is really, this is an initiative grounded in deep purpose, authenticity, listening and collaborating with people, a recognition that we can do better, and a willingness to collaborate broadly. And my last segment here is we polled the CSO leadership community and asked them to complete a survey in which they provided 3 words that best represent the work, values, and community of Common Sense Oncology. This work comes from our CSO graphic design consultant and game show host, Professor Chris Jackson. And like any good academic, before I share the results, I have to tell you about the eligibility and exclusion criteria. They were asked for 3 words. So we had a few comments. Bishal needs a haircut, 4 words. That one was removed. Someone was trying to make him look extra good for Netflix. And there's another statement, Booth should retire and focus on his Bollywood dance career. And that comes from, again, I'm preserving anonymity here, but a well-known thoracic surgeon in Mumbai who appears to like my dance moves, but is not very good at counting because there was 10 words replied instead of 3. But in all seriousness, this is the word cloud that came. And I think it was, really gratifying to see these are the words that our community identify this initiative with. Patient-centered, equity, inclusive, compassion, value, honest, welcoming, and yes, sometimes we can be a little bit provocative. So thanks again. Looking forward to kicking off the next 3 days of meeting. I'm now going to hand over to my younger brother, Bishal, who comes here from Nepal to Japan, to Harvard to Kingston, and then direct from Hollywood to introduce our keynote plenary speakers. Thank you very much.
So thank you very much, Chris. And I would like to welcome you all again to Montreal, to the snowy Montreal, and also people who have joined us online for this plenary session. It's our distinct privilege and honor to be inviting 4 distinguished speakers for the plenary session today. First, I'd like to introduce them all very briefly. You all have this document in your file folder, so you can go through this for the detailed introduction. So to save time, I'm going to do a 1-sentence introduction for all the plenary speakers for today. So we have Dr. Monica Bertagnolli. She was the 17th director of the NIH in the United States, the first surgeon and second woman to hold such a position, and also the 16th director of the NCI and a former president of ASCO. Thank you very much. We are really honored and privileged to have you here. Then we have Dr. C.S. Pramesh, who is the director of the Tata Memorial Hospital and professor and head of thoracic surgery at the Tata Memorial Center, Mumbai. Thank you very much for joining us. We also have Henry Scowcroft, who is an award-winning science writer and author of Cross Everything, a memoir about the loss of a loved one to cancer. And he also is a member of the BMJ's International Patient Advisory Panel. Thank you very much for joining us. And last but not the least, we have my favorite, Professor Eisenhauer, who is my mentor. And she was the first director of the investigational new drug program of the NCIC clinical trial group, now known as CCTZ. And she has won almost every award that is there to win in Canada and internationally as well. We'll hold off Q&A until the very end. So thank you very much.
Oh, geez, thank you so much for including me in this meeting. And I can't tell you how wonderful it is to be among international colleagues who really think strategically about what we need to take much better care of our patients, to make life much better for our patients. While there's so many exciting new advances in biology and medicine, they arrive every single day. And we as clinicians know we are just scratching the surface. Decades of investment in fundamental biology have produced insights that are now translating into really exciting new approaches in disease prevention, treatment, diagnosis at an unprecedented rate. But there is so much more to do, and everyone today here knows that effective clinical translation remains incredibly challenging. Long COVID, Alzheimer's disease, early cancer detection, autism, long-term effects of environmental exposures like microplastics. The 30 million people living in the United States alone who have a rare disease, 30 million people, and more. Each of these yet unmet challenges needs so much more and could benefit so much from more clinical research. And in our health systems, we pride ourselves on delivering evidence-based medicine, but it is very sobering to consider what kind of evidence clinicians and patients have available to them to make life-altering decisions every day. You all know this study. I'm sure you've seen it many times. It's 6 years old, a reprise of a similar analysis 10 years before. And sadly, the situation today is no better than it was 16 years ago. Although our clinical guidelines get bigger and bigger, the level of evidence we have to guide care, the needle doesn't change. And we really have a level of evidence much lower than we'd like. And add to this issue the fact that clinical trials evidence we all believe in passionately when you're making life-altering decisions involves carefully specified cohorts, frequently leaves out people who are the majority of those receiving care, such as children, older adults, and people with comorbid conditions. Now, of course, we don't need randomized trials for every decision. The famous parachute study reminds us of this. But there is a lot to be done in the space between simple observation, and it's perfectly obvious, and randomization of highly selected cohorts that can bring so much benefit to the clinicians and patients in clinics every day. And even healthcare practices based on parachute-like assumptions are falling away because of new approaches. For example, remember when it was absolutely necessary for a patient to come in and see a clinician every single time they needed a health assessment and a treatment planning visit. No longer what we do. In 2011, the National Academy of Medicine in the US released a consensus report detailing requirements for achieving precision medicine's goals of improving healthcare by treating the right patient with the right therapy. 2011, we thought we'd be cracking along at this very quickly. A fundamental concept articulated by this group was the need to develop think about disease differently, develop a new taxonomy that allowed a merger of clinical information with data from fundamental science. Well, those of us working in clinical trials were already doing this at the time in pursuit of new biomarkers, which I think have made some progress. But a decade after delivery of the human genome, this was also a much needed call for more collaboration between discovery science and clinical research. And it was a foundational document also for the concept of what we call a learning health system. The term learning health system was first conceptualized in 2006 during a workshop organized by the National Academy. This concept built upon earlier ideas around evidence-based medicine and the recognition of gaps between biomedical research findings and their application in healthcare delivery at its first iteration. Here's the definition of learning health system that I like the best among many: an integrated, important word, health system, which harnesses the power of data and analytics to learn from every patient and feed the knowledge of what works best back to clinicians, health professionals, patients, and other stakeholders to create cycles of continuous improvement. I think that about covers what we need to do. And so much has changed over the almost 20 years after this vision was first articulated, but the building blocks for evidence generation by a learning health system remain exactly the same. Foundation of high quality digital data or computable data, the ability of researchers to access the required data, and tools and analytics that allow us to derive insights from data. And you all know it has been much more difficult to assemble this infrastructure than we anticipated. I could take an hour more to detail the many challenges, but in a nutshell, the reason that we do not yet have the learning health system that we seek is that perverse incentives, many perverse incentives in our healthcare environment have prevented us from having access to the data that we need. So one of the perverse incentives has been that held us back is that health systems and health information technologies have allowed data to be a commodity that, for all intents and purposes, has health system intellectual property associated with it, essentially limiting access to most researchers, cutting patients out of the loop, and not allowing data to be used, interrogated at scale across our very diverse national healthcare systems. This is in spite of the fact that for at least a decade, we've had all the technology we need to drive the learning health system. We do not have a learning health system because of perverse public policy. It's not because we don't know how to do it or we don't have the tools. Okay, well, ultimately, data used to achieve the aims listed here. We need data to achieve the aims listed here. Every clinician and patient will have the evidence needed to guide health decisions. Health systems will drive optimal outcomes while maximizing efficiency and minimizing cost of care. And individual people will know how their health data are being used and can participate in research that's meaningful for them when they wish to do so. I'll give you an example from when I was at NIH, which kind of blows my mind that here's where we are in 2025. We had studies for gene therapy for little boys with Duchenne muscular dystrophy. We needed to find them between ages 2 and 3. And it was a race because there were kids with this horrible disease who were going to turn 3 years old in a month. We needed to find these kids and get them in and find them fast. Absolutely impossible. No way to find them. And it's just frankly obscene that we did not have the ability to do that. We had to count on patients coming together and providing their registries, or clinicians who focus. This is not the way to serve people. After 20 years of calling for this, we still don't have an effective learning health system today. Another area where we have been held back is by thinking that a learning health system is only about the so-called real-world data to be used for quality initiatives and health outcomes research. Not at all the case. We, as clinical trialists, use data every day. Where does it come from? It comes from the clinical care environment. And I will tell you, I am no fan at all, having been deeply involved in knowing what, quote, real world data really is, including in countless validation studies, tracking it back to the source doc and realizing that, I'll tell you, roughly about, you get a real world data set and you compare it to manual chart review, it's, of all those data elements you see in that data set, about 50% or less are going to be actually an accurate demonstration of what was really going on with the patient. It's awful. I haven't found one yet that is a collated data set that is much better than that. Clinical trials are an outstanding use case for building a learning health system instead of the kind of bad data we have now. Clinical trials demand rigor, scientific rigor in design and execution. They produce results that change clinical practice and can provide the evidence base for marketing new products. Evidence from clinical trials could be dramatically improved by features that a learning health system would deliver, such as more comprehensive and longer follow-up. Again, in 2025, there is no need to lose a patient to follow-up because they moved from Boston to Chicago. Doesn't ever need to happen. We could also examine outcomes throughout the natural history of the disease. Think about following a woman with metastatic breast cancer through every single line of therapy with overall survival as the outcome, even if that woman has participated in 6 or 7 or 10 clinical trials. We can absolutely do this. And then finally, and this is really important for us in clinical trials, generalizability studies. Okay, fine. We have our specific cohorts. We're trying to ask a really rigorous scientific question. But then when we let that out into the wild, we really ought to be able to know what happens. And that knowledge from what's happening in the wild, the generalizability when the drugs and therapies get into real use, is incredibly valuable, and we can do that. And then last of all, there's our data environment. Whenever I give kind of a talk about data, I say, Amazon knows what kind of shampoo I use, right? And it's shocking, on my cell phone, I went to Los Angeles last week, and the minute I land in Los Angeles, there's like 10 little ads for where I can go to get a bagel, right? We can also have a real-time data interaction with our research clinicians and their patients. There's, again, no excuse for this. Well, there's lots of reasons why. I'll get there. Okay, the mechanism of achieving adaptable, fit-for-purpose data collection required to achieve a learning health system would also facilitate more efficient and effective clinical trials. Meeting the needs of the spectrum of study types listed here by an integrated data information system would definitely achieve the integration we seek between research and clinical care. What do we do today? Okay, here's a rough look at what we do today. According to a study protocol, raw data at a site is formatted according to specifications set by the study. Very rarely are the data pulled directly from the EHR. Even if they are, they're already kind of pulled over into some other formatted thing. You never ever get raw data, of course not. And then it's de-identified and then sent to a central aggregator. And geez, we stratify based on site. Why? Because again, we've looked. Boy, the data is different from site to site. And so, of course, we'll never stop stratifying, but believe me, a lot of that bias can be removed if we look at raw data. Let's see, what else? We pull all this data across many sites. It's checked for completeness and consistency. And if a registry study comes, then now one electronic data set is compared to another electronic data set by people who physically go there and look at the two on site. Really kind of insane if you think about it. Finally, the curated, aggregated data is finally cleaned many, many, many months later and used for research. In some, I'm making the extreme case, I know, but this happens still far too often. Well, this is incredibly slow. It's expensive. There's loss of data fidelity that occurs when many individuals are translating data into standard formats. Back and forth interactions between the clinical care and data management team are laborious and slow and do not allow prompt corrections. And it is difficult to train and maintain. This is probably the biggest incentive for change. It is very difficult to train and maintain machine learning assists among so many different types and different approaches. Because although, yeah, that's supposed to homogenize, diversity, that's not what it does when it's working the way we need it to work. So we need to do better. It doesn't need to be this difficult. There are simpler ways that not only better serve clinical trials, they utilize an infrastructure that's central to building a true learning health system. And we need technology to be used in the right way to do this. About 8 years ago now, with funding from the FDA, which was the big incentive because the real world data mandate for FDA was there, it was on the books. FDA is supposed to use, quote, real world data for regulatory decision making, and they were freaking out, frankly, and I don't blame them with how bad the data were. So we started doing projects with the FDA about getting data that really would pass regulatory muster for a regulatory trial out of the electronic health record. This was a collaboration done with MITRE and the Alliance for Clinical Trials in Oncology. That was the start of the collaboration. It's gone on significantly since then to build an infrastructure required for clinical trials that would fit the objectives of the learning health system. The goals are here. You can see them. Use the EHR both to identify study populations and to collect data. Integrate research data collection with routine clinical care in a minimal burden manner. Allow patients to communicate more effectively with researchers and clinicians, and make sure that data collection included diverse populations. Above all, the design features had to be rigorous enough to allow FDA approval intent trials to be conducted. So here's a simplified description of one approach that has been achieved that we can demonstrate meets all of these requirements. Again, this is simplified. You're going to see there are lots of details you probably like to hear, but I think you'll get the point. Start with the data user. Data user, in this case, are the researchers conducting the trial. They request specific data to meet the needs of their study. They don't ask, and it's been hard for us to teach people to do this. They don't ask, what data do you have? Or what data can you get? Because every time they ask us that, we throw it back at them and say, what data do you need? Don't ask us what data we have. Tell us what data you need. It's been shocking how difficult that is for researchers to make that shift. Anyway, so what data do you need? And then the team works with them to develop a customized study schema that gets integrated into the EHR and into clinical workflows. And it's a raw data request to all participating health centers. Raw data. The middle, that's a broker. That is not a data lake. It is not a data holder. It's just a data transmitter. So the data lives at the sites. The broker identifies ways to pull it in and pulls it in to meet the specifications of the clinical trial the user. All links to raw data are maintained, greatly simplifying the quality checks. You don't check source docs. You have the source docs. It's the raw data, but then gets formatted as to however the researcher wants it. But all the links are there. Daily interactions between the brokers and sites ensure completeness. Curated data sets are readily provided to researchers or DSMBs as requested. We had a data set of 18,000 patients that the team, in response to a new request, this wasn't a trial, it was a new request for data on patients with lung cancer, 18,000 patients. The team delivered the data. It was a specified data set in 3 weeks, completely done, all tracked to raw data. Cost and time to data completeness are dramatically reduced. Technology required to achieve this is available and already deployed in some settings. The architecture in this example works because it works from raw data captured centrally, tracks the lineage of all data elements, and maintains provenance and perpetual record of all data location and sites rather than itself holding data. This also allows the creation of a universal health record for a patient as long as if they're at any site where this capability is introduced. It enables quantifiable and rapid approaches to maintaining data quality and completeness, as well as rapid generation of requested data reports by either sites or users and even patients. This is one approach. This is what the technology can do. I wish I could tell you we've got this adopted at scale across the country, but I can tell you this is very real and has been demonstrated and is happening today among collaborators. We know we can do this. It is now our job to make sure that technologies that make this possible now contribute and are rolled out in ways that actualize, not detract from a learning health system. We made that mistake already with EHRs. So let me end this slide with a slide that details the tenets of the learning health system as envisioned by the National Academy of Medicine in the US. You will see that all of the attributes that this fabulous organization has been talking about in just different terms, but the concepts behind all the work done by these teams are also listed as part of the learning health system. They are essential for moving clinical trials operations forward to achieve faster, more comprehensive acquisition of the evidence we require to benefit patients in society. So thank you very much again for allowing me to be here.
Thank you so much. Maybe we'll take just one question, so we don't want her to be surprised. So maybe just one question. The most burning question.
I'm sorry.
Not extremely burning, but wondering if the system you set up captured images or only numeric,
Oh, no.
alphanumeric.
So the system literally can capture anything. Not only can it capture again, the capabilities are all there. It is very limited deployment across basically 4 health systems right now. It can capture any data. It captures image files. It captures medication distribution from CVS pharmacies. It captures utilization data. It captures raw data. And these programming teams and data science teams can map that raw data to whatever specification a user wants. So, and then track it back down if the need be. So the data can go wherever it needs to. I will tell you that there's a lot of work being done right now with industry partners, places like CVS, places like insurers. And before I left HHS, a program to do exactly this for federal health data reporting called Data Counts, which we're waiting to see if the program continues to go forward. But the teams that have built this are absolutely adamant that federal program will have a counterpart in a nonprofit space to be able to use for the benefit of the learning health program.
It's not enough for what we need. So how did you guys manage it to harmonize syntaxes and taxonomy? Otherwise, it's like barely can get 30% of that.
We work with the raw data. And the raw data is mapped, but that's what you're doing today. Okay, think about it. You're taking raw data, a CBC, an imaging report, whatever, and you're mapping it into a case report form. Any mapping can be done. It's just the raw data is moved to the mapping. If you want to map it to OMOP, whatever other real world data type set or algorithm, it can be mapped. The important thing is, which is very different than the way data from the clinical care environment is handled today, is that you can map it any direction, but you can always, if you have a question, track it back down to the source. And that's the big key. Because you're right, there's no mapping that's perfect, ever. None of our clinical trials data sets are perfect. But when you need to, you can always track it back. And this was one of the essential features that we built in to adhere to the requirements that FDA challenged us with back in 2017. All right, well, enjoy the rest of the meeting. I'm glad I got to be here with you all. Thank you.
So without further ado, I'd like to invite the next speaker, Dr. C.S. Pramesh.
Thank you, Bishal. Great to be back among friends and amongst people I know so well. When Chris asked me to speak about the National Cancer Grid and reducing inequities, I had to think a lot because I knew Ian, Richard, and Chris would be here, and it's probably the 17th time just in 2025 that they've heard me speak about it. So I had to try and spice this up a little. And given the fact that it's almost 4 o'clock on a not so great day, I thought I'll spice things up with a question. How many of you believe that this statement is true? The world has never been a better place to live in, even today, given current situations. Okay, so the majority of you who don't believe this that's the default. When this question was asked to 20,000 people, 90% of those who live predominantly in high-income countries disagreed with the fact that the world was probably in the best situation that it could ever be in. And I'll tell you why most of you were wrong and most of these 20,000 folks were actually wrong. If you look at many of the metrics that we use to track how the world's been doing, whether it be a human development index, you see uniform increase in human development index regardless of which region you belong to. If you were to look at GDP, it's growing up variably in multiple locations, but nevertheless going up in all locations. Extreme poverty is dropping drastically. Child labor is dropping. Legal slavery, just 200 years back, practically every country in this world had slavery legalized. Today it's zero. Food supply adequacy going up in practically every location in the world. Polio almost eliminated in most parts of the world. Childhood deaths under the age of 5, reduced by more than 90% in just a couple of centuries. Education levels are going up, average years of education going up uniformly across the world. Global literacy rates growing up again. Total number of oil spills from tankers at an all-time low. Death rates from natural disasters, all-time low. And even today, people living in a democracy, and I've taken 4 50-year circles. And just to take you through this, the red ones are the bad ones. The red ones are autocracies. The shaded gray are where we don't have the data. And the blue ones are the best. And this was 1823. Fast forward 50 years, 1873. 1923, where practically most of the world was ruled by autocracy. Even 50 years back, most of the world was still ruled by autocracy. And a dramatic change in the last 50 years, where predominantly we live in countries where there is access to democracy. We might see one of those dark circles, blue countries change color this year, but nevertheless, it still remains the fact that at no point in history have we had more people living in a democracy than we have today. Paradoxically, and this is the paradox that we need to understand, rarely has the world been more inequitable than today. And I'll explain this. If you were to take overall income into account, the top 10% of the world's wealthy account for 52% of the world's income. And the bottom 50% account for less than 10% of the world's income. If you were to stretch this to overall wealth, the top 10%, it becomes even more stark. The top 10% account for 76% of the world's wealth, and the bottom 50% account for 2% of the world's wealth. And contrary to what you might imagine, this discrepancy between the haves and the have-nots is less marked among high-income countries as compared to low and middle-income countries. If you look at sub-Saharan Africa or the MENA region, you see the maximum discrepancy between the haves and the have-nots. And this becomes even more stark when you look at overall wealth, where if you look at countries in Latin America and MENA, just under 2% is the entire wealth of the bottom 50% of the population. And I did show you these graphs, but they don't tell you the full story. So if we were to look at what the current Human Development Index in Sub-Saharan Africa is, it's what it was in OECD countries 150 years back. Though there is improvement in these regions, there's still a long way to go. Similarly with GDP, what you have in Sub-Saharan Africa was what OECD countries had 200 years ago as their per capita GDP. And unfortunately, we know that healthcare expenditures are almost directly correlated to the per capita GDP of a country. The richer the country, the more the expenditure on healthcare. And again, though this is not universally true, there is a rough correlation between how much money you spend on health. There are some exceptions. I don't want to get into the nuances of that, but predominantly, if you spend more money, your people live longer. Which is why we need to think of this from a global perspective and to think about what this means for cancer care across the world. And I'll use the example of India because I find India is more a microcosm of what's happening in the rest of the world. This is a heat map of 75,000 patients who visited us at the Tata Memorial Hospital. And this means that patients are willing to travel thousands of kilometers to access what they think is high quality care. And this doesn't even take the wealthiest Indians into account. They probably go into high-income countries to get cancer care. The projected burden in countries like India is expected to go up dramatically. In just 2 decades, we expect a 60% increase both in cancer incidence as well as cancer mortality. And by 2040, 70% of all cancer deaths will occur in low and middle income countries. This is just an evaluation of India, measuring the gap between what resources we have now and what would be ideal even to take care of patients with cancer today. And what you see here is approximately a 1.4 times to a 2.6 times increase in resources just to meet today's demands, not adjusting for the projected requirement in a few years. And this is probably again a reflection of what is happening in many parts of the world. So 12 years back in India, we had to think of something quite disruptive to address this problem and to try and catch up with the gap as much as we could. So we created what we call the National Cancer Grid, which was a loose network of existing cancer centers, which were looking at how could we create uniform standards of care and narrow the gap between the high-performing centers and the low-performing centers. And over the last 12 years, we've expanded quite massively. We are currently at close to 400 cancer centers, research institutions, patient groups, and professional societies across India, which between them treat 850,000 new patients with cancer annually, approximately 60% of all of India's cancer burden. So when we thought of looking at how could we address this issue of promoting equity in cancer care, we needed to be strategic about how we use our resources. So we came into 4 main areas that we thought our efforts should be directed towards. The first was on the quality of care, and we had some efforts towards that. The second is to address the issue of access and affordability. And again, we have a few initiatives in that. The third was research and innovation. And we felt strongly that this was one area which we could use to our advantage to overcome many of the problems that we've been facing in cancer control in similar parts of the world. And finally, we felt the responsibility to create what we call NCG Global, which is looking more outwardly and trying to address inequities which are shared between many other low and middle income countries as well. So the first aspect of quality of care, we try to address with resource stratified guidelines. Again, something which has been embraced by many other countries in the world, thanks to the fact that our guidelines are much more implementable as compared to those guidelines developed in high income countries. And again, this has been endorsed by the National Health Authority in India, which links reimbursement to the largest public health scheme in the world to adherence to these guidelines. The second aspect of quality was diagnostics quality, something which is not so glamorous in today's world. We all look at sexy cutting edge technology like robotic surgery, proton therapy, and immunotherapy, forgetting the basics of diagnostics and diagnostic pathology, where quality leaves a lot to be desired. So what we did was to create a web-based lean version of the CAP accreditation, which looks at H&E staining, immunohistochemistry, and more recently, molecular pathology. And we didn't stop with just doing a quality assessment. We had these runs of quality improvement projects where at any point when a weakness was identified amongst some of the diagnostic facilities, there was educational activities which were taken to make sure that these weaknesses were overcome. So this is estrogen receptor positivity, progesterone receptor positivity. And over the years, you can see the improvement in performance. This is HER2 new testing for breast cancer. And just to take you through, the blue bars are good, the well-performing centers, the green bars are not so good. And as you can see over the last 5 years, you see a steady increase in the number of blue bars. The second bucket of access and affordability, we address through virtual tumor boards. We've been doing this for close to 10 years now, over 1,200 virtual tumor boards, more than 6,000 patients who have been discussed here. And it's also been a great resource for case-based learning, automatically improving the quality of care in many of the smaller centers. The heat map demonstrated to you how far patients are willing to travel just to get an expert evidence-based opinion. So what we did was to create a web-based platform wherein patients can upload reports from any part of the country and increasingly across the world. And this goes to a backend of a software company that we work with who convert it into a structured format, sends it to an expert within the cancer grid who then feeds back an opinion, all happening in a median turnaround time of 24 hours. Over 110,000 such opinions have been offered, 65 countries, and surprisingly, out of those 65 countries, 23 were actually high-income countries, including the US and Canada. demonstrating that access to high quality evidence-based decision making was not a problem confined to low middle income countries. The final bit about access and affordability was an initiative that we took for group negotiation for cancer medicines, given the fact that most of the newer cancer medicines are extremely expensive and out of reach. We aggregated demand from multiple centers, pooled their volumes, and negotiated with industry, getting discounts ranging anywhere from 23% to 99%, with an average of 85% discount on the cost of these medicines. This is a study done by one of Chris's fellows, which looked at 700 randomized trials in cancer. And to me, this slide gives me three clear messages. First is the graph on the left. There's virtually no correlation between cancer mortality, which if we could use as a surrogate of cancer burden, and the top randomized trials which were performed in cancer. The second was the fact that 92% of these randomized trials were led out of high-income countries. Given that 70% of cancer mortality will happen in low-middle-income countries, this seems extremely skewed. But to me, dangerously, 87% of randomized trials tested systemic therapies, which means that between surgery, radiation oncology, palliative care, survivorship, and practically every aspect of cancer control that you could think of, we just accounted for 13% of all trials that were performed. So therefore, we felt the need to establish and have prioritization of what our cancer research priority should be. I think practically half the authors in this paper are probably sitting in this room, but thanks to an intense brainstorming activity, we were able to identify 5 key research priorities. And I'll just leave it on for half a minute just for you to go through them. And I want to emphasize here that if you look at these priorities, none of these are probably priorities for in high-income countries. Probably none of these are certainly priorities for the pharmaceutical industry. And given the fact that most trials in today's world are funded by the pharmaceutical industry, these important priorities get sidelined. So the other aspects of research that we wanted to address was the lack of research training. So we address this through our own equivalent of the Whale and the Films and the Accord workshops. We run this as an annual event, which has become extremely popular now. So far, we've trained over 500 early career researchers from 32 countries, increasingly becoming international, including biostatisticians and more recently patient advocates as well, just to ensure that they are well trained in how to contribute towards clinical research. So this was the latest version, which we had last month. Many of the folks in this room were faculty at this meeting, and more than 40% of the participants were from outside of India in other low-middle-income countries. And I will take this opportunity to thank many international organizations, including the US NCI, the IAEA, St. Jude, City Cancer Challenge, and the WHO for funding many of these participants from low and middle income countries. So what happens in this intense 6-day workshop is that participants come in with a 1-page research concept, which is a fairly vague idea at the back of their mind, and over a period of 6 days gets converted into a full-fledged research protocol ready for submission to ethics committees. The final hurdle for research in LMICs is funding. So the National Cancer Grid funds research which aligns with those 5 priorities that I just mentioned. And these are all, some of these trials have actually got completed. This is a childhood ALL trial, which completed recruitment in over 2,200 patients, looking at repurposing drugs, cost-effective treatment interventions, problems which are specifically common in low-middle income countries. These are head and neck squamous cell cancers. Again, aspirin getting repurposed as a anti-cancer agent, post-mastectomy, radiation and N0 early breast cancer. The Hematology Cancer Consortium, which has recruited over 36,000 patients as part of a very large conglomerate of centers within India. Just demonstrating 2 important features. If we work together, we can actually get very high recruitment in large trials in low middle income countries and done at a fraction of the cost of what it would take to do the same trial in a high income country. Going by all of this, we wanted to centralize our efforts in digital oncology, hoping that these digital tools would help us overcome many of the barriers that we face as researchers. So foundation gave us funding for setting up a Center for Digital Oncology, many of the points which Dr. Bertagnolli spoke about in a previous talk, including interoperability between electronic health records. How can we aggregate data seamlessly between varying health records, as well as an increased focus on AI and ML in cancer research. The final part that I want to speak about is NCG Global, which is a more recent phenomenon. This was announced by in the International Atomic Energy Agency General Conference a few years back, just before COVID. We were slowed down a bit because of COVID, where we were practically inactive as far as the global component was concerned for 2 years. But now we've increased. And in the last annual meeting of the National Cancer Grid, we had over 250 leaders from cancer centers, including ministries of health, and leaders of cancer centers from 24 countries from various parts of the world. And as part of the last annual Cancer Grid meeting, we had 2 meetings, one with the ASEAN, which is the Southeast Asia network of countries, and the BIMSTEC, which is the Bay of Bengal initiative, both of which were aligned to sharing best practices from the National Cancer Grid with those countries, and we've already started working with them closely on that. The WHO Southeast Asia Regional Office also requested us to start a Southeast Asia version of the Cancer Grid, which again got launched earlier last year. And 40 member organizations from 11 countries from the Southeast Asia region are now part of the National Cancer Grid. An effort of this nature wouldn't be possible without the support of several national and international organizations, many of whom are represented in this room, and I'll take this opportunity to express my deepest gratitude to all of them. Thank you very much.
Thank you very much. We'll park all the questions for the end. So we'll have our next speaker, Mr. Henry Scowcroft.
Thank you very much, everyone. I'm delighted to be here speaking to you all. My name's Henry. Got a very different type of talk for you in that I'm not going to be talking about great big initiatives or amazing national infrastructure. I'm just going to be talking about my story, my journey to be speaking here in front of you. Yeah, it's just wonderful to be here in front of you all. So thank you very much for having me. These are my grandparents. This is Mari and Edmund. They're why part of the reason I'm here is thanks to them. My grandmother was a medical illustrator. She illustrated biology textbooks. My grandfather was a plant biochemist. Discovered all sorts of crazy things about plant respiration. But as a young boy, they fascinated me with the world around them. That led to me going to university to study biochemistry because I thought I wanted to be a scientist. I got there, I realized I really loved the science. I was hopeless at doing it, but really enjoyed talking to people about it. That led me to do a master's in biochemistry, a master's in science communication, sorry. The reason I'm telling you all of this is because as this story unfolds, there'll be all sorts of things that have come along the way that map very closely onto some of the values and the aims that CSO has and that we all have as a community together. So Masters in Science Communication taught me all about how to talk to non-scientists about science, which is something I was absolutely determined to do. I thought maybe I wanted to be a journalist, but that looked like a somewhat difficult career of mainly criticizing science rather than evangelizing about it. So my first job after university, after my master's, was with an organization that will be familiar to many of you. It was with Cancer Research UK. And I stayed at Cancer Research UK for nearly 2 decades. The main achievement I had while I was there was I started at Cancer Research UK in 2003, when the internet and its role in the communication of science was not what it is now. And myself and 2 other colleagues had this idea that maybe we could write a blog, which at the time now seems like a really archaic thing. Blogs very, very 1990s almost, but at the time for a mainstream charity to be writing a blog was quite a new thing. So we launched the Cancer Research UK Science blog, and I like to think that I owe my career to the Daily Mail getting things wrong a lot because they gave us a lot to write about. And we would write on a daily basis about the sorts of things that were making the headlines about cancer because we wanted to really convey a lot of the media comments that we put out as a charity would say this research is interesting, but more research is needed to confirm its findings. But actually, the research was interesting, and we wanted to explain what was interesting about it and what research was needed to confirm its findings. So we were able to write on a daily basis on the internet, relatively unfiltered, as unfiltered as you get working for a big organization, about what was going on in cancer science. And that led in 2009 to winning an award for this. And the chap to my left in that is a guy called Ed Yong, who went on to be a Pulitzer Prize winning journalist for The Atlantic. We had some good discussions in the early days of this blog. But anyway, we were writing about cancer on a pretty much daily basis for a good decade or more, and quite a lot happened between the years of 2004 and 2014 plus in cancer. But as well as writing about those things, we broadened out towards just writing about the science. And this was one of the first big hitters that we ever published. This was a decision by the National Institute of Health and Care Excellence, the HTA assessment body in the UK, to say no to some kidney cancer drugs that had just come out. And we had the wacky idea at the time of just saying to people, what do you think? What do you think of this decision? And we had nearly 100 comments, which the charity then pulled into our submission back to NICE. And I won't claim the blog caused this or led to this, but the outcome of this whole saga was that the NICE produced the end of life care guidance, and this blog played a small but important part in that story, and it gave us a clue that we were onto something with this whole internet malarkey. But mainly these were the things that happened that really stuck with me over that time writing for this blog. You had the arrival of targeted therapy, and I think we've all seen this gruesome and yet weirdly hopeful picture of vemurafenib completely eradicating all the tumours in someone up melanoma and up torso, and then them coming crashing back later. And this idea that we're getting somewhere with the biology, but we're not getting where we need to get. Then we had the arrival of immunotherapy in about 2014. Suddenly, we saw overall survival for immunotherapy, the thing that had been worked on for decades, finally started to work. But this was the story that really, the third thing here is the story that really captured my imagination, with tumor evolution, and building on Mel Greaves' work, Charlie Swanton's work in 2012, 2013, looking at the branched evolution and tumor heterogeneity. And this really started to me to really explain some of these features of cancer and how it all fitted together. I interviewed Charlie several times for the blog. I worked with him on the launch for some of the papers that came out about this. And I think it's fair to say, but by 2016, I had a fairly conventional view of the biology of cancer. This is the well-known hallmarks of cancer. You're all very familiar with this, but the idea that cancer can be broken down into several discrete different hallmarks that combine to produce cancer, the disease as we know it, as we all know it. So up until 2016, my story was very much a kind of media-driven, biology-driven view of cancer. And that all changed in 2016, when my partner at the time, and I would put up a picture of her, but I think this graph almost tells the story more. She was diagnosed with stage 4 bladder cancer. The survival, 1 year survival, 29%, median survival time about 9 months. And suddenly, for the first time, cancer came into my life, not as a biological entity with hallmarks or with branched evolution or with Kaplan-Meier curves, but as an actual clinical disease, but actually the real human aspect of living with cancer. And I won't go into all the details of the subsequent months, but the first thing that I did with my reductionist view of cancer was I phoned up Charlie Swanton and said, is there anything you can do? So he did we got a biopsy over to him, and he had a look for the weak points of the tumor. There were none. The therapy at the time was cisplatin. That was about it. We knew that all cisplatin would do would potentially buy some time, but we knew that immunotherapy was on the horizon. My partner's best friend herself had melanoma and was responding incredibly well to pembrolizumab. So we knew that even though there was no hard outcome data for immunotherapy in bladder, we could see someone who was benefiting from that. So we knew that an immunotherapy trial was what we really wanted to go. And so the large focus of our lives became trying to get onto an immunotherapy trial. And I'll come back to a minute to talk about the process of enrolling on a trial, because everything we've just been talking about in the last two speakers' talks so resonates. And some of the points I want to make are absolutely the points that we made in a different way earlier. But yes, average survival time, 9 months. Turned out, they always say don't believe the statistics, but it was bang on 9 months to the day. So after all of this, I thought, what the hell do I do about all of this? I've known cancer for a decade and a half, and now I've known it for 9 months in 2 totally different ways. How can I bring this together? So I did what all communicators do. I wrote a book. I wrote a book about the human experience of it all, but I also wrote a book that was about the cancer itself. And we took the sequencing data, we took the biopsy data, the various blood samples donated to UCL Biobank. We put it all together and we told the story of the tumor. It arose from a single clone of cells which diversified into two different types. One went on to create another. You can see there the light blue is the original founding population. The dark blue and the green were the branches, and then the dark blue clone became the green clone. You can see how, if you're familiar with these fish plots, you can see how the chemotherapy eradicated certain types of the tumor, certain clones within the tumor, and then it grew back again after the therapy response. And you can see the biology and the human experience absolutely mapping onto each other. Where do I go with that? What's next? Yes, the trial. So this was the trial that we tried to enroll on. There were the frustrations, I'll come to this in a minute, about trying to get on this trial. There were bureaucratic hurdles, there were clinical hurdles, all the kind of things that a learning health system that we just heard about should hopefully get us around. But ultimately, my late partner passed away a couple of weeks after initiating therapy on the trial. So at the end of all of this, I came to a very different view of cancer, very similar in its appearance, but these are the hallmarks of living with cancer. Abnormal health, changed priorities, uncertainty, loss of control, new vocabulary, but also importantly, hope. So that was the whirlwind 9 months of 2016. Today, I'm still doing comms. I've left Cancer Research UK. I moved on to Alzheimer's Research UK for 2 years. Now I'm an independent consultant. I'm working with CSO on some of the communication strategy. We're going to be talking a bit more about that on Thursday's session. I'm a writer and editor. I sit on the BMJ's patient advisory panel. I also sit on the management group for a couple of early phase cancer trials. And I've been working with some academics at LSE on some interesting research around the communication of the benefits of cancer drugs as well as the risks. But putting all of that together, and also the important bit as well, I wear the hat of being a dad. Hannah and I recently welcomed Robin into the world. He's a lovely little 4-year-old, and I'm missing them terribly as I stand here in front of you all. But yes, some thoughts on drawing all of this together. So, trial enrollment. The experience that Zara had trying to enroll on the Biscay trial was fraught with hurdles. The trial took ages to get open. And then we had this extraordinary process where, in order to be eligible for the trial, despite having measurement after measurement taken as part of our routine NHS care, we had to do a whole load of new measurements for the trial enrollment, which necessitated the one extraordinary thing where her hemoglobin had to be a certain level. So we were basically rushing into hospital every 2 days to be filled up with more bags of blood to try and get to some artificial level to enable her to get the trial drug. And it struck me at that point that there was a charade going on here, that ultimately everyone wanted the trial to go ahead and to her to receive the drug, but there were these arbitrary hurdles being put in the way. And everything that the first speaker, Monica, talked about in terms of a learning adapted health system were to get around all of this stuff, being able to find a way to find the patients who are eligible to the trial, take their existing data and pull them into the trial like that would speed things up, but also avoid all of that sort of back and forth. This is a really interesting one. Chris and I have talked a lot about this in terms of endpoints. We get really overall survival is absolutely vital to measure, but all of the other endpoints that I feel that sometimes in CSO, sometimes we're a bit dismissive of them, but they're all important to someone in some situation. Because ultimately, when you're sitting there in the oncology consultation, you have questions and you want the answers to them. Some of the endpoints are the answers to those questions. What are my chances of responding to this drug? That's response rate. How many people are going to have a durable response? PFS, DFS. How long is it going to give me? Overall survival. But how will it make me feel? Problems. All of these things are really, really important. And I feel occasionally we get so fixated on on banging the drum for overall survival that we forget that all of these metrics are important in their own way to different people in different circumstances. As I phrased it here, one person's surrogate is another person's primary. Absolutely behind the work you guys are doing in terms of quality of life and PROMs. I sat on a TMG recently where I was banging the drum to make sure the trial was going to collect quality of life measure data, and the PI said, "Yeah, but we don't really want to be asking patients about how they feel. That's going to remind them how awful they feel, isn't it?" So after having won the battle to say, all right, we're going to collect PROMs, they said, all right, so which outcome metric, which PROM score, which PROM survey do you recommend we should use? No one knew. We had no idea what the best way of measuring quality of life was, even once I, as a patient advocate, had convinced the trial group to measure it. So we really need to nail this one so that it's really easy to do this, because it's what certainly many of my advocacy colleagues bang the drum in TMG meetings or in the trial design meeting. Got to measure this stuff. How? What's the best way? But I think the drug trials need to do things for different groups. They do need to give industry the data so that their effective drugs can get approved rapidly, and that rapidly, when you're living with metastatic advanced cancer that's moving very, very quickly, the speed with which this stuff coming through the system really matters. So absolutely agree that we need to make sure that bar is high, but we can't sacrifice the speed as well. That's really so important. You need to give clinicians that information on what are good options. You need to provide current patients with answers to their need, but you also need to provide patients to the future with better drugs than we have now. These are, there's so many different things to balance here. Also, trials that matter. We heard earlier that 87% of therapies of trials are looking at systemic therapies. What about other things? When my late partner was struggling with lymphedema, and we were told there were 3 different treatments: there's compression stockings, there was lymphatic draining massage, or there was diuresis. And we said, so which is the best one? Don't know. No idea. Why hasn't the trial been done? These kind of pragmatic and simple trials should be done. But also, as I'm sure we're going to be talking about the next day or so, therapy duration, dose de-escalation, really important stuff. Involving patients and carers with all of these decisions, and from the very sort of upstream stuff about research strategy, what questions are we going to ask, to the trial design, to the information materials that are given out on trials. It's so important. But I also want to make the point as well that research isn't just about clinical trials, and taking part in research as a patient isn't just about clinical trials. This is a study that came out a few months after my late partner died. It's a study that her cells were involved in. It was looking at lymphocytes from the urine as predictors of the immune response, immune microenvironment in the bladder. She didn't enroll on a trial for this, but she took part in the research. And this is just as, well, I won't say just as important as getting access to a new therapy, but in terms of getting out of bed in the morning and knowing that you're doing something with this awful thing that's happening to you, taking part in research and knowing that your data is being used to make the world a better place is so important. So yeah, in conclusion, then wrapping it all up, I think you guys are doing vital work through CSO. It's on every level, the equity, the improving rigor in trial design. Overall survival is important, but I do just want to make sure that in your messaging and the way you talk about these things, that the other endpoints can be important too. Don't undermine the way these things come across, because actually knowing that my tumor is going to be stopped growing, knowing that I might respond to this drug is important as well, because cancer care is so much more than trials. Improving it is so much more than trials. It's all of the other stuff as well. It's the biological research, it's the palliative care stuff, all so important. But anyway, thank you very much for listening to me.
Thank you for the excellent talk. We'll move ahead with the last talk of the plenary session. Dr. Eisenhauer.
So thank you very much. It's a great honor to follow 3 such tremendous lectures. I was thinking about all the things we have in common, the work we do that brings meaning to others, but also to us as individuals, and the growth that the work that we do provides to ourselves. And what I'm going to do in this last talk of this afternoon is really talk about some of the lessons I've learned over the course of my career as a clinical trialist and as a human being. No disclosures. So in this talk, I'm going to cover two broad areas. One, lessons I've learned from being involved in clinical trials. And the second part is really lessons I've learned from the patient groups and patients that I've been involved with. And then at the end, really talk a bit about how CSO can help this area. So let's start with my work at the Canadian Cancer Trials Group, some stories and lessons, but first a bit of history. The National Cancer Institute of Canada Clinical Trials Group was formed in 1980 with funds from a charitable source, the Canadian Cancer Society. And the National Cancer Institute of Canada was the research arm of the CCS at that time, and they decided to establish a clinical trials group emulating some of the successful work done in the US around cooperative groups. And Joe Pater was the inaugural director of the group and remained in that role for more than 25 years. And here's a picture of Joe back in the day. For those who know him now, maybe you'll see some familiarity. Now, at the time, the academic mission of the CTG was articulated, and it really remains the same to this day, to develop and conduct trials with a goal of reducing morbidity and mortality from cancer. To do this, three key components were required: the scientific and clinical leaders from whom ideas were generated, a network of cancer centers and hospitals that would be the place where trials would take place and patients would be recruited from, and a central statistical and coordinating center that was based at Queen's University, where we worked. And in 2016, the NCIC CTG was renamed the Canadian Cancer Trials Group, and I'll use both names interchangeably as I jump around a bit of history. In 1982, the Investigational New Drug Program was created, and the reason for this goes back to how cancer drugs were developed at that time, although it's kind of hard to credit in today's environment. In the 1970s and 80s, pharma was not really interested in developing cancer drugs. It was seen as too high risk, and a huge gap was therefore filled by the US National Cancer Institutes in their drug development program. But Canadians had kind of poor access to the drugs that were coming through the system at that time. So in 1981, a committee was formed by Joe to look at how we might be more successful. And the recommendation was that an oncologist be hired to work in the central office to help identify new drugs in the pipeline, collaborating with our US colleagues, and put in letters of intent so we could access some of those drugs for study in Canada. So in 82, the Investigational New Drug Program was founded, and there I am, much younger, and anybody in the room who's into fashion recognizes this is the 80s by looking at the shoulder pads on that jacket. In the early years, our work really focused on a number of different agents, most of whose names have been long forgotten in the annals of history because they weren't very effective. But I want to highlight one in particular. This trial, IND number 22, was a trial of deoxycoframicin in a rare leukemia called hairy cell leukemia. And this was a trial based on work by James Johnson, a hematologist in Winnipeg, who studied it in a lab setting and found it to be very active. And in our phase 2 trial, our amazing response rate was 100%, 89% complete response. And interestingly, the very first patient enrolled on that trial was by me in Kingston, a gentleman who'd just been diagnosed at the time the trial was open, had a massive spleen and marrow infiltrated, and after only 3 weekly doses, had a normal spleen and a normal marrow. And herein lies lesson 1. As an investigator, as a human, I can tell you there's nothing more exciting than participating in research that has such a profound impact on an individual's life. Sadly, it doesn't happen all that often, but when it does, it's unforgettable. The 90s were called the taxane decade for good reason. Taxol, an extract from the bark of the Western yew, was found to be active actually way back in the 1960s in the NCI screen. But everyone thought its mechanism of action was just like that of vincristine, widely used and available, so it wasn't really looked at until Susan Horowitz in 1979 showed it had a unique mechanism of action, and that prompted a fairly rapid movement to formulate it and move it into clinical trials in the US. And those early trials had some significant problems. Arrhythmias, severe hypersensitivity reactions, likely due to its formulation in cremophore. And under some circumstances, those observations would have allowed it to be abandoned, except already in phase 1, it was looking like it was producing tumor regressions in patients with ovarian and breast cancer. So a lot of work around how it was given was put into place. Pre-medication was added, infusions were prolonged, patients were admitted to hospital for monitoring, and so on. And in Canada, we were interested in looking at this drug and thought, well, what can we contribute? And that led to us designing what was called the OV9 trial, which was my first foray into randomized trials in ovarian cancer. And this particular trial asked the question of how do we optimally deliver this drug? In a relapsed ovarian cancer, we randomized patients to 2 different doses and 2 different schedules of administration in a bifactorial design. And we found that the shorter infusion, somewhat surprisingly, was actually safer, but still effective, and the higher dose was more effective. And that really changed how the drug was given to this day. But almost as important, or maybe more importantly, 2 other developments came from that work that we'd done with colleagues from Europe and Canada. The first was sort of obvious. We then went on to design a collaborative frontline trial, this time led by the EORTC, looking at frontline use of paclitaxel in ovarian cancer. And you can see the results there, improvement in overall survival of a median nearly a year. But it also started us thinking about collaborating more broadly between Canada and other groups themselves were starting to get excited about that activity. So out of this grew the Gynecologic Cancer Intergroup. You can see a nice picture in a beautiful resort in Germany there of representatives of over 30 cooperative groups around the world that are now together working on gynecologic cancer clinical trials. And on a personal level, of course, I met many colleagues who became lifelong friends. So my second lesson, collaborations across borders and oceans not only speeds clinical research, which is important, but it creates lifelong friendships, which are as important as we work in areas like this. So moving ahead another decade, the first targeted agents appeared in the 2000s. Of course, these drugs were focused on trying to interrupt the signaling pathways that seem to be important drivers of cancer. And an early example I'll give you from the CCTG experience, this one not my own trial, but my colleagues, focused on the epidermal growth factor receptor shown here in this cartoon, often overexpressed or mutated in epithelial cancers. And so an early area of interest in developing inhibitors. And CO17, a trial we did in Canada in collaboration with the Australian group, was the first randomized trial of an EGFR monoclonal antibody in colorectal cancer to show a survival advantage, as you see here. So cetuximab in this case improved survival compared to best supportive care. And this trial also, because it collected tissue, was able to reveal an important biomarker in terms of RAS mutation status for patient selection. Now, I don't have time to go over all of the things that happened since. Suffice it to say that the CCTG and international partners have undertaken now many, many trials of targeted drugs, angiogenesis inhibitors, immune therapies, many of which have now given new standards of care. But important to remember that there are some trials that only academic groups are interested in and only academic groups could do. And we've had a fair share of those. This recently published example is one, a surgical trial comparing radical to simple hysterectomy in early cervix cancer, showing, and Marie Plante did this trial. She's an investigator from Quebec City, and in collaboration with the Gynecologic Cancer Intergroup, similar cancer outcomes, but of course, more complex surgery and more side effects in the radical group. A trial published a decade ago by Tim Whalen, again, with international collaboration in the US, UK, Australia, looking at regional node irradiation in early breast cancer, no impact on overall survival, though higher lung toxicity and higher local recurrence rate with the radiation. Short course radiation plus temozolomide or radiation alone in glioblastoma, a trial done with EORTC, showing improved survival in the temozolomide arm. And finally, a trial called CO21 or the challenge trial, a structured exercise program being prescribed to patients in one arm of the trial, post colorectal cancer surgery versus standard of care. And our very own Chris Booth is co-principal investigator of this trial. And stay tuned because results are expected imminently on this trial. So the third lesson, pharma and new drugs alone will not be able to address all the important questions in cancer management. So academic trials groups really are vital. I'd like to sort of pause for a moment to acknowledge the Canadian Cancer Trials Group and Janet Dancy, who's here somewhere. See her, is the director of the group today. And you can see there are numerous sites across Canada that are members, and we continue to this day to be supported by the Canadian Cancer Society with individual trials, also supported by US NCI, CIHR, and others. So just summarize the first part, academic groups, I think, are critical in studying new treatments, and these trials have improved outcomes in numerous cancers, and those that we've done in Canada have really been enabled not just by the national network, but work with international collaborators. But as we know, cancer remains a considerable problem with a major cause of premature death internationally and suffering, despite these strides. So we are not by any means done yet. So changing gears, patients as partners now. This is a term that's used very often these days, and we have to remember it wasn't always this way. Patients were seen as research subjects in the 1970s or 80s when I started my work, but that really has changed dramatically over the last 2 or 3 decades. The idea that patients can play an important role in clinical trial design and peer review is one that's now widely adopted. And in our own group, first Helen Gent and then Judy Needham have led really remarkable groups of patient volunteers as part of our representative committee. But I'd like to really focus on 2 other things that have come up already in the discussions, and that's how patients can play important additional roles in defining the endpoints of greatest meaning to them and questions for trials themselves. So let's talk about endpoints. As we know, historically, phase 3 trials utilized endpoints that might be considered by many patients as the most inherently meaningful, that is overall survival or cure rates. And in the last 20 years, that's changed quite a bit with the rise of progression-free survival as the primary endpoint. And this graph, Chris has presented at least once today, maybe more than once, showing the increase in the use of progression-free survival as the primary endpoint in the last few decades in high numbers of trials in the solid tumor types shown there. It's useful to reflect upon what progression is, because it isn't inherently intuitively obvious. And progression, as we use it today, dates actually back to the island of Corfu. Now, that sounds a little strange as I say it, but in 1995, there was an early drug development meeting in Corfu for the EORTC, the NCI, and the NCIC-CTG. And as we were listening to phase 2 trial reports, there was a hallway conversation where we realized that different cooperative groups were starting to modify criteria to suit their needs or what seemed reasonable. And there was getting to be a bit of a drift in how we defined patient outcomes in phase 2 trials as a result. So as a result, we put together an international working group to try to standardize and address this. And this work eventually culminated in the publication in 2000 of the response evaluation criteria in solid tumors, the so-called RECIST criteria. Now, a key part of those criteria was the progression definition. And that is not a single thing, it is a composite endpoint. We picked arbitrarily, for some good reasons, a 20% increase in tumor burden or tumor size, or the appearance of new disease, or non-equivocal, whatever that means, increase in non-measurable disease, or if it's used in a randomized setting, death. And it was intended that this endpoint be used as one of 4 categorical outcomes in non-randomized phase 2 trials, the others, of course, being complete response, partial response, and stable disease. And even with that definition, what happened to patients who met the definition could be drastically different. And let me illustrate that here with a cartoon. This is an example of, let's say, a patient who has a small nodule in their right lung, and on treatment, it grows by 20%. And so that met the PD criteria. But equally, this is progression, a dramatic increase in tumor, new disease, a pleural effusion. And you might imagine that the forward trajectory of what happens to those patients, both meeting the PD criteria, would be drastically different. So not too surprisingly then, when you actually look at whether a progression event or a time to progression like those 2 different things, it may not be a good marker of how long someone may subsequently live or what they feel. So I won't say it's never a surrogate, but almost never a true surrogate of overall survival. And it doesn't correlate very well with what happens to quality of life in many publications. Nonetheless, PFS is being used for a variety of reasons. We know why. It moves things forward faster. It seems to be easier to measure, although some might argue with that. But a lot of people have argued that it's an important endpoint for patients. But no one had really asked them that. And that was something that we thought we should do. So with Mike Brundage's leadership, we set up a trade-off type experimental design in a multi-center fashion and asked 100 patients with metastatic cancer what increase in time to progression would lead you to take a new treatment that had some toxicity, but that had no impact on your survival chances. And this was published a year ago. And as is often the case in these types of experiments, patients had a variety of views. About 17% said they would take the new treatment even if there were 0 months gain in PFS. About a quarter said they would take it for some gain, anywhere from 3 to 9 months. But just over half said they would decline the treatment no matter how much PFS gain there was, given that it didn't affect survival and it didn't affect, and it had some toxicity. And the point of this is to say there isn't one answer, but rather that prolonging control of tumor size is not universally valued by patients. So using that as an argument to be measuring PFS seems to have some weakness. In a complementary findings from this recently published study by Forrest, patients were asked about how important it was to have quick access to a drug and survival benefit. And their key results just highlighted here. Patients, not surprisingly, would very much like to have some certainty of survival benefit in making a decision about treatment, but also they were very much against having long delays in getting hold of that treatment. But they did say that if they had a very low certainty that a drug would benefit them in terms of survival, they would be willing to wait, or they would rather wait. And you can see the median time of how long they'd be willing to wait was close to 2 years for strong evidence of certainty. So my 4th lesson is patients we need to listen to because they can provide important guidance that will shape trial design and endpoints, and there will not be a single answer for every circumstance and every patient group. Now, what about identifying questions for trials? Because we all know that, as I've indicated, many cancer trials have both clinician and patient input, but I guess where I was really educated was how sometimes patients can raise questions that clinicians would not normally consider. I'm just going to show you one example from the US, a myeloma example. And in 2002, at that time, myeloma treatment was amongst other things comprised of using high-dose glucocorticoids, dexamethasone, and you can see 600 milligrams of dexamethasone in a 28-day cycle, huge dose of drug with a lot of side effects. And Mike Katz, who was a myeloma patient on the ECOG Akron Myeloma Committee, pointed out that patients wanted freedom from the side effects of this drug, and could we not just do a trial where we tested lower doses? Now, this was a time when all sorts of sexy new things were happening in myeloma, and the investigators wanted to look at new drugs, but his reasoning prevailed, and after some debate, a randomized trial was designed looking at lenalidomide plus the standard high-dose dexamethasone plus a lowered versus the same lenalidomide dose and a lower dose of dexamethasone. That trial was published in Lancet Oncology. David's going to think I'm doing some advertisement for him here. And you will see, small font, but you can see Michael Katz is one of the co-authors, the patient whose idea this trial was. Not surprisingly, the lower dose on the right column had a highly significantly reduced serious toxicity or early mortality, but unexpectedly, the lower dose also had a significantly improved overall survival from treatment. So lesson 5, patients can and should be defining trial questions. So just to summarize, academic clinical trials can make vital contributions to improving cancer outcomes. We will not fight our way out of the burden of cancer treatment and the needs of cancer patients simply by new drug development. Patients are integral not just as trial subjects, advocates, research reviewers, but they can also guide us as to what the best endpoints are for the questions we're asking and be a wise source of ideas for trials. And so my final lesson is common sense oncology. What can it do in this regard? I think you can help us ensure that academic trials continue not just to survive, but thrive, and underscore that patient input remains front and center in determining the questions worthy of study. So I thank you for your attention.